Welcome to this explainer. Today, we're doing something really exciting. We are going to dive deep into the foundational elements of behavioral neuroscience. But we aren't just going to look at a list of dry facts, right? We're going to treat this field exactly like what it truly is, the ultimate scientific mystery. I mean, think about it. How on earth does a 3-pound mass of wet, biological tissue inside your skull generate your entire reality? To solve a mystery this massive, to really teach all things foundational to this incredible field, we need to understand exactly how the detectives of the mind have built their toolkit over thousands of years.
So we have to start with a question that totally baffled early civilizations and basically launched our entire investigation. Where does the mind actually live? Now, it seems incredibly obvious to us today, but for a very, very long time, humanity was looking in completely the wrong place. Here is our roadmap to teach you all things foundational for this field. We're going to cover the mind-body mystery, the blueprint of the brain, the 5 levels of analysis, the neuroscience toolkit, and finally, the future of discovery.
Okay, let's dive straight into Section 1, the mind-body mystery. For centuries, brilliant minds debated where consciousness actually sits, and honestly, their theories were wildly off base. Take ancient Egypt, for example. The heart was considered the true center of the soul and memory. In fact, they held the heart in such high regard that they actually thought the brain was useless. They just discarded it during mummification, literally pulled it right out through the nose. Fast forward to ancient Greece. You've got Hippocrates arguing that the brain is the center of intelligence. But then Aristotle completely disagreed. He thought the brain was basically just a biological radiator designed to cool down the fiery passions of the heart. By the time of the Roman Empire, a physician named Galen realized that nerves connected to the brain, but he thought they worked by pumping these invisible animal spirits through hollow ventricles. It literally wasn't until the 19th century that scientists finally figured out nerves were actually electrical wires and not, plumbing pipes.
Now, this history represents a massive philosophical shift. Back in the 17th century, René Descartes popularized dualism. This is the idea that the physical body and the non-physical mind are completely separate things. It's essentially the idea of a ghost hovering in the machine, communicating, or so he believed, through this tiny little structure called the pineal gland. But modern neuroscience subscribes to monism or materialism. This is the understanding that the universe consists only of matter and energy. So under monism, your mind isn't some magical spirit floating around in your skull. It is a physical phenomenon produced entirely by the intricate biological workings of your nervous system.
Moving right along to Section 2, blueprint of the brain. Behavioral neuroscience is heavily guided by Charles Darwin's principles of natural selection. Because, look, the human brain didn't get this massive and powerful by accident. It was forced to adapt to survive. Think about the intense selective advantages that required so much neural complexity in the first place. We developed agile hands to meticulously craft tools. We developed color vision to spot ripe fruit hiding in the canopy, or, predators lurking in the brush. Bipedalism, walking upright, that freed our hands entirely for long-distance travel. And then came the absolute heavy hitters. Linguistic abilities allowed us to share collective generational knowledge. And then there's the social brain hypothesis. This suggests that your massive cortex evolved specifically to help you navigate the incredibly complex relationships, alliances, and politics of everyday human social life. And that brings us to a core, truly mind-blowing concept in modern biology, neuroplasticity. The brain is absolutely not a hardwired computer that comes off an assembly line with fixed permanent connections. It is highly plastic. It's constantly changing, adapting, and remodeling its very architecture based on your daily experiences. It can even produce entirely new cells, a process called neurogenesis, well into adulthood. Seriously, think about that for a second. Every single time you learn something new from an explainer like this one, your brain is physically rewiring itself in real time.
Okay, Section 3. 5 levels of analysis.
So, with an organ this incredibly adaptable and complex, how do neuroscientists actually break it down and study it? Well, they use a highly interdisciplinary hierarchy. Picture zooming a camera lens in and out. At the very bottom, zoomed all the way into microscopic levels, you have the molecular level. That's studying the unique chemical messengers of the brain. Take one step up and you're at the cellular level, looking at how those molecules give individual neurons their special properties. Then we zoom out to the systems level. Here, we're studying how massive constellations of neurons wire together to form circuits for things like vision or hearing. Zoom out again to the behavioral level, analyzing how those systems produce integrated actions, like an animal foraging or navigating a maze.
Finally, zoomed all the way out to the big picture, we have the cognitive level. This is studying how all of this brain activity creates the mind and higher-level mental activities like imagination, language, and self-awareness.
To make sense of it all across those 5 levels, our scientific detectives have to perform a bit of a balancing act between 2 opposing approaches. Going back to our detective metaphor, reductionism is like looking really closely at the individual threads at a crime scene. You're explaining a highly complex phenomenon in terms of the tiny elementary processes that underlie it, like studying the firing of a single muscle cell to understand the entire act of walking. Generalization, on the other hand, is zooming out to look at the whole tapestry. It's about forming a broader explanation based on lots of smaller observations. For example, deducing the evolutionary role of a specific brain chemical by watching how it affects aggression across a whole bunch of different mammalian species. A truly great neuroscientist is constantly toggling back and forth between these 2 perspectives.
All right, Section 4, the Neuroscience Toolkit. Let's see what's actually in the box.
The most obvious hurdle in this entire field is just a daunting anatomical one. I mean, the brain is encased in thick bone, so how exactly do you figure out how to peer inside a living, thinking skull without actually harming the person? Well, today, we divide our non-invasive tools into 2 distinct categories, structural and functional. Think of it like the difference between studying a computer's hardware versus its software. Structural imaging tools, like CT scans and MRIs, they act like incredibly high-resolution anatomical cameras. They show us tissue density and physical pathways. That's the biological hardware. But if you want to see the software running in real time, you need functional imaging. PET scans track radioactive tracers to measure metabolic activity. fMRI tracks blood oxygen levels as a proxy for neural firing, basically watching where the blood rushes to feed the active parts of the brain. And MEG goes even a step further. It detects the minute magnetic fields produced by active neurons to give us temporal resolution down to the millisecond.
Now, imaging is amazing, but it's largely correlational. It shows us what areas are active during a task, but it doesn't necessarily prove they cause the task. For those deeper causal studies, researchers turn to animal models using the oldest method in neuroscience, experimental ablation. By carefully destroying or even temporarily turning off a specific brain region and then observing the behavioral changes, they can figure out exactly what that region does. But here is a really crucial point for scientific accuracy. Researchers absolutely must use sham lesions. Think of this as a surgical placebo. They perform all the exact same steps of the surgery on a control group, the anesthesia, the incision, everything except the actual tissue destruction. This rigorous control is the only way to ensure that any behavioral change is caused purely by the missing brain region and not just the shock of the surgery itself.
Beyond simple ablation, the lab technology we have today is absolutely staggering. It borders on science fiction. With a technique called clarity, scientists can extract the light-absorbing fats from an animal's brain and replace them with a water-soluble gel. This literally renders the brain tissue completely transparent. They can see deep structures without ever having to slice the brain. Then there's Brainbow, which uses genetic manipulation to make individual neurons express hundreds of different fluorescent colors. It visually untangles the complex wiring like a glowing neon map. And my personal favorite, optogenetics. Scientists actually use genetically modified viruses to insert light-sensitive proteins into specific, targeted neurons. By just shining a blue light through a microscopic fiber optic cable, they can artificially turn specific neurons on and off. They are literally controlling behavior with targeted beams of light.
And that brings us to our final section, the future of discovery.
One of the major holy grails of modern science right now is mapping the connectome. Think of this as the ultimate comprehensive wiring diagram of the human brain, aiming to reconstruct every single synaptic connection. The sheer scale of this data is almost incomprehensible. We are talking about mapping roughly 86 billion neurons and upwards of 100 trillion connections. Massive global initiatives like the Human Brain Project in Europe and the Brain Initiative in the US are using vast networks of supercomputers to simulate the brain and trace these dense, functional highways that essentially make you who you are.
But look, with tools this incredibly powerful, as we map trillions of connections, read magnetic signatures, and manipulate cells with lasers, the ethical guardrails become absolutely paramount. Animal research in this field is strictly governed by the 3 R's. Replacement with non-animal models whenever possible, reduction of the absolute number of animals used, and refinement of procedures to minimize distress.
For humans, strict institutional review boards and the absolute, unyielding necessity of informed consent protect volunteers. And on top of all that, the rapidly emerging field of neural ethics is working overtime to navigate the implications of all this data. This is especially true regarding cognitive privacy, because, let's face it, our scanners are getting better and better at effectively reading minds. Yet, despite all our pristine anatomical maps, our glowing genetic fluorescent hues, and our massive supercomputers, we always arrive back at the ultimate unsolved mystery, the hard problem of consciousness. We can watch a memory form in real time on an fMRI. We can trace the precise neuropathway of physical pain. But we still don't fundamentally understand the translation point. How exactly do purely physical, chemical brain processes generate the subjective, internal, vibrant feeling of what it is actually like to be alive? So, we leave you with this final, profound puzzle. We know the molecular parts, we've mapped the intricate cellular systems, and we've tracked the deep evolutionary history. But how do these simple biological materials conjure the miracle of your unique thoughts, your deepest emotions, and your entire subjective reality? The detective's toolkit has never been sharper, but the greatest mystery of the mind is still waiting for the next generation of detectives to solve it. What will we discover next?